welcome back again. Just looking at a blank screen this time. Giving your mind a minute to rejoin us. Hi and hello again. Genetics, reminding you that while we are probably pretty familiar with genetics, we're just going to touch on critical concepts that we're going to be discussing a lot as we're moving forward. And also that when we talk about genetics, this is the actual machinery of inheritance. And remember that the only thing that matters when we're talking about evolution are changes, things that are inherited, things that change from generation to generation and are inherited. Remember, we have to have a link between the genotype and the phenotype, and between the phenotype and fitness. Just thinking about human beings, we have really extraordinary variation. Even if we're just thinking about eye color, this is a really easy example. We have all shades of green, all shades of like brown to gold to very, very dark brown, all shades of blue to like green blue, colors that are almost violet, extraordinary variation. And then if we take humans as a whole organism, wow, massive amounts of variation. And even if we just take things that are super simple and easy to pick out, like height, like hair color, hair texture, the shape of someone's cheekbones, wild amounts of variation in our species. And these examples that I'm using I'm using them because they're familiar, because we are humans. But this is just variation in our species. Every species has this extraordinary variation. And to remind you that all of this variation comes from our genome. Our genome, which is made up of genes. And I'm going to touch back on this in a minute, but remember that each of these chromosomes in this karyotype over on the left side of your image is made up of DNA. And we can find variation. So this little example is just kind of showing you all across the human genome in so many places. All of this phenotypic variation that we see comes from extraordinary genotypic variation. Massive, massive, massive amounts of genotypic variation. So thinking about the human genome, we have 3.2 billion base pairs. Each of our chromosomes, let's go back here for a moment. Each of our chromosomes has about a million base pairs. Each chromosome. That's a lot. Each chromosome has about a million base pairs. And if you're not 100% remembering what that means to base pair, we're going to go over that in just a second. So our entire genome is about 32 billion base pairs, which is pretty cool, I think. Among organisms on Earth, we're not particularly big or particularly small. Some bacteria have a genome as small as 20,000 base pairs. And there's a salamander called an axolotl that has 32 billion base pairs in their genome. Remember, we have 3.2 billion. So this axolotl salamander has 10 times the number of base pairs in our genome. And you may remember from genetics that the size of the genome does not speak to an organism's complexity. Humans, we're just kind of here hanging out in between these extremes. So we are 3.2 billion. An axolotl is 32 billion base pairs. We're talking about base pairs. And then bacteria Not all of them, of course. We can get down to 20,000 base pairs in their entire genome. Discussing genome, when I'm talking about the genome of all the organisms that I've talked about so far, we are of course talking about DNA, deoxyribonucleic acid. And hopefully you remember from genetics that DNA, deoxyribonucleic acid, is a double helix with a sugar phosphate backbone. So this sort of cool little loopy thing here that I'm outlining in white is the backbone. Sugar phosphate backbone. It's made of a sugar and a phosphate, which is why it's called that. And then what sort of look like the rungs, if we're going to follow the ladder analogy, are the bases. And so when I talk about base pairs, I'm talking about the bases. So we have adenine, which is represented by an A, always pairs with thymine in DNA. So this is a base pair. So over here on our DNA, if this was adenine and this was thymine, this would be a single base pair. Adenine always pairs with thymine, and you probably remember guanine always pairs with cytosine. These are our base pairs. So these 4 bases make up the extraordinary variation that makes us. The extraordinary variation in the many, many, many different types of phenotypes, not only in humans, but in all life on Earth. All life on Earth uses these 4 bases to make it what it is, which every time I think about that just stinking blows my mind. Okay, so the vast majority of what we're going to be talking about throughout the semester, we're going to be focusing on what's happening with DNA. It's important to note, particularly now, that there are some viruses that don't use DNA, they use RNA. So there are some viruses that their genetic material is not DNA, their genetic material is actually RNA, and one of those viruses is the coronavirus. And so this image, I just thought this was really cool, and an important thing about these viruses, these RNA viruses, is that they can't replicate themselves. They have to come in to a host and co-op our systems, co-op our replication system, what our body does to make our RNA and our proteins, and it hijacks that. It hijacks that system, and what it does is it hijacks that system to make more of it. So if you look in this process, we've got our virus, which is sort of represented by this little circle thing with the squigglies in the middle and the little yellow things poking off. And what it does is it comes and it binds to one of our cell surface receptors. So those receptors, which are the lock, the viruses have the key. So they come and they bind to one of our receptors. And by binding to one of our receptors, what that does is that allows them to inject their genetic material into our cell. And then that genetic material instructs our cell to go through this process of translation. So it starts with translation. You're probably thinking, wait, what happened to transcription? Well, we're starting with RNA, right? So we go from RNA, translation, and it translates these little proteins, makes a whole bunch of these little proteins. And then these proteins transcribe another RNA. Then it goes through this whole process. We go through transcription, and then we go through more translation, and then all of these different RNAs get translated, and here it makes all the little parts of the virus. So it makes all the little parts of the virus, which is an endoplasmic reticulum. Then the virus RNA, which is right here, gets wrapped up into that coating, that endoplasmic reticulum, goes through the Golgi apparatus, then goes through exocytosis out into the rest of our freaking body. So our own system gets hijacked by RNA to make viruses. Important side note, cool. Not everything uses DNA. So going back to DNA, because we're going to be talking a lot about humans. We're also going to be talking about mostly not about viruses. Just reminding you the way this works, we have our DNA base pairs over here kind of in the bottom that I might be covering up. I'll go this way. Okay, so we have our DNA base pairs down here in the bottom. If we kind of zoom out of our DNA base pairs, we have introns and exons, which I'll talk more about in a minute. All of that gets coiled tightly around histones, and then even that gets coiled even more tightly into chromosomes. If we go back and we think more about how do we get from DNA to protein, because remember, protein is the functional units of our body. Protein is what actually does work. Proteins are generally what produce our phenotype. We start with DNA, and then we go to pre-RNA. So we go to pre-RNA, and the process of going from DNA to pre-RNA, remember, is called transcription. And then to go from pre-RNA to RNA, we have to go through a process where we take out, I don't think I marked them on here, introns. So the introns are in orange. We got to take out those introns. Remember, we process that RNA. We take out the introns. We very gracefully, much more gracefully than my image here, put those exons together to form a functional or a mature RNA. It leaves the nucleus, goes through a process called translation, remember, at the ribosomes, and we get a protein. That's just an overview. We went through that in a ton of detail in genetics. If any of those points are unclear to you or confusing, I suggest you go back and review what's called the central dogma of genetics. So that's our genetics overview. Next thing we're going to do is we're going to overview mutations.